Good morning, everyone. I am very glad and honored to introduce our distinguished speaker, Dr. T Shivaraman. Dr. Shivaraman has finished his BSc and MSc in chemistry from Bharathidasan University, Tiruchirappalli. In the year 1999, he has received doctoral degree from the Department of Chemistry, National Tsinghua University, Taiwan, with a specialization on structural biology. Later, he has done PDA from College of Medicine, University of Iowa, USA, and worked as postdoctoral fellow and research fellow at Department of Biological Science, National University of Singapore. Before joining Karpagam, he has also served in the Department of Biochemistry, Ponnaya Ramajayan College, Tanjavur, School of Chemical and Biotechnology, Shastra University, Tanjavur. Since 2017, he has been associated with the Department of Biotechnology, Karpagam Academy of Higher Education, involving in both teaching and research. Dr. Shivaraman has 83 publications in reputed journals with a cumulative impact factor of 141. Also 3 books, 2 book chapters, and 1 patent to his credit. He has more than 100 presentations at various scientific events. Sir is chairing editorial board member and reviewer of many reputed journals. He is also life member in various associations like Indian Association for Cancer Research. Dr. Shivaraman is invited for research collaboration by Axiohelix Private Limited, Japan, and also as resource person in many conferences. He has also received travel grant for his research work. Sir is an active member in the Academic Council, Karpagam Academy of Higher Education, Coimbatore. Focused research area of Dr. Shivaraman is protein chemistry, its structure, folding, and functions. And he has many more achievements in his credit. With this short introduction, I can surely say that he is the right person to talk about today's webinar on the topic computational tools for molecular modeling and docking simulations. I wholeheartedly welcome our speaker, Dr. Shivaraman, to take over the session. Have a knowledgeable session to all of you. Thank you. Over to you, Dr. Shivaraman, sir.
Good morning to all of you. First of all, I express my sincere thanks to Dr. Mohan, Principal, Karpagam College of Pharmacy, and Dr. Senthil Kumar, the organizer of the event, for giving the opportunity to share some of my research works with all of you. And also, thank you so much for the nice introductions, Dr. Asha Madam. Let me start the topics. So the title of presentation, Computational Tools for Molecular Modeling and Docking Simulations. Before getting into the topic, just 1 or 2 minutes, I would like to discuss about the research works right now we are doing in my laboratory. This slide is my academic path, starting from BSc to professor at present. As you can see, all my degrees, BSc, MSc, PhD in chemistry. So I have been faculty members in the Department of Biochemistry, Bioinformatics, and Biotechnology. And these are my research areas. I have classified my research areas into 5 different fields. The first one is structure, function, relationships of snake toxins. That is toxinology. The second research area, drug designing by computational and as well as experimental methods. At present, mainly we focus on cancer biology, then molecular dynamics and simulations, developing novel algorithms, and nanochemistry and biology. These are some academic metrics of mine. Okay, I move to the next slide. This is my official website. If you are leisure and if you are interested, you may please visit to the website, and the website provides a lot of computational tools. So I have listed the computational tools over here. The computational tools are very useful to address some of the frontline research problems in protein folding. all the tools are freely available and accessible. Okay. And I hope everyone see the slides. Okay, now I'm showing the research outcomes. So in this slide, I have mentioned some of the research achievements the lab has done so far. As I mentioned in the previous slide, we have developed novel computational tools, which are very unique to address some of the frontline research problems in protein structure and protein folding. Our lab has developed novel anti-cancer compounds. So we have designed a de novo anti-cancer compound. The compound has been validated using the in vitro as well as in vivo experimental methods. So we have also developed antibacterial compounds against dengue virus, antiviral compounds against leptospira. Leptospira is the bacteria that causes the leptospirosis. We also developed the anti-venom compounds. Actually, our lab is the first one to develop inhibitors to cardiotoxins, which is the principal protein toxins in snake venoms. So we have developed the first inhibitors to the cardiotoxins of the snake venoms. So these are the few research outcomes of my laboratory so far. And what I like to say is, irrespective of the diseases, irrespective of the research problems you have, given the target, our lab is full-fledged to screen as well as design the compounds to the target by using an array of computational strategies. Okay, so let's move on to the topic. Okay, so you can stop me. You need not to wait till my presentations. If you have any genuine questions, you can please unmute and you can bring the questions to me, or you can post the questions in the chat through the Dr. Sendil Kumar. I will address the question.
Okay, sir.
Okay. Sir, is it okay, sir?
Yeah,
Everything
is it
fine?
okay only? Yeah.
Okay.
In between, there is a disturbance won't be there means then we'll grant, sir.
Okay, sure.
Okay.
Okay, let us start the topic today. Very simple topic, very interesting. Let me try my best. As far as consider computational biology, all the works in one way or other are relevant and as well useful to the drug design. So now let us start with the drug design. So all the computational works, no exception at all, are very useful for drug design. Okay, so I think all of you know very well. So what is drug? Any student just unmute and tell me. Okay, I answer. So what is drug? What do you mean by drugs? So drugs are the small molecules or biological macromolecules having potential to cure or prevent disease or to enhance mental and physical welfare. I think the definition holds good. Okay. Okay. So, okay. Now we understood what is drug. Now, the drug designing process, as we all know that, is a highly complex, expensive, and time-consuming procedure. A recent statistic suggests that there are about 10 to the power of 60 chemical compounds right now. Just imagine 10 to the power of 60 chemical compounds. So those 10 to the power of 60 chemical compounds need to be screened in order to identify the lead compounds. So from the lead compounds, we have to identify the drug likeness compounds. From the drug likeness compounds, we have to identify the drugs. Okay, so make it simple. What do you mean by lead compounds? Lead compounds are the compounds that are entering into the preclinical studies. So drug likeness compounds are the compounds that enter into the clinical studies. So what are drugs? Drugs are the molecules that enter into the market. So from the compounds, we have to identify the lead compounds, drug likeness compounds, and drugs. So it's a very highly complex process, of course, and very expensive process, time consuming also. Okay. Now, the drug designing. Okay, when you come to the drug designing can be carried out either by pure experimental methods or by pure computational methods. Now, our focus for this presentation, computational drug designing. Okay, irrespective of the method, whether experimental methods or computational methods, for drug designing, we should know the two informations: the structural information of target molecules, structural information of small chemical molecules. We need the structural information, right? So where we can get the structural informations? For small molecules, as you can see in the slide, the structural informations can be obtained from the chemical databases such as PubChem, NCI, Zinc, and so on. Okay. For macromolecules, the structural informations, the structures of the macromolecules, in other words, can be retrieved from protein databases, PDB. Okay. Now let's see where the molecular modeling and molecular talking come into the picture. Now we understood what is drug, what are the complications in the drug designing. For drug designing, what are the prerequisite parameters? What are the prerequisite parameters? Structures of the small molecules, structures of the macromolecules we need. Okay. Let me move on to the next slide. Sir, is it okay, sir? Dr. Siddhil, sir?
You can proceed, sir.
Okay, sir. Okay.
If you have any doubt, students or delegates, any doubt, unmute yourself. You ask, because this is an interactive sessions. Kindly unmute it, then ask your questions.
Okay. Okay, let me move on.
Yes, sir.
Okay.
You can start, sir.
Okay, sure. Now in this slide, see, I'm showing the various screening strategies. To become a drug, the compound, as we all know that, right, should successfully pass through the various screening stages. So the affinity stages. So what we are looking for, the affinity between the target molecules and the ligand molecules. And the now specificity. The drug molecule should be highly specific to the target. Then we look for bioavailability and as well as toxicity, PK/PD properties. So all the stages, all the major 4 screening stages can be carried out by experimental methods. Now what I'm telling is all the 4 stages can also be carried out by computational methods. So that is the interesting part of the computational biology, right? Without touching the experimental, all the 4 stages can be carried out by computational biology. So now you understand the importance of computational biology. So the computational biology is very, very useful to speed up the drug designing process, to make the drug designing very smart. We need the computational biology. Okay. Now, when you say the drug designing, there are a lot of drug designing strategies. So just for your student benefit, so I have mentioned the popular drug designing methods, like structure-based drug designing. This is what everybody talking about. That is, of course, very important, but there are so many. High-throughput virtual screening, de novo drug designing, ligand-based drug designing. Just for your sake of clarity, I'm just reading. Fragment-based drug designing, core hopping, peptidomimetics. And you can see one more interesting point, peptidodiamimetics. This is a drug designing strategy our lab developed. So there are a number of strategies, right? Each strategies are unique to address a certain problems. So there is no one drug designing strategy. Depending on the question, depending on the problem you have to choose the right drug designing strategy to bring the best answer. So what is ultimate aim? We need a drug molecules. Okay. So these are the number of drug designing strategies. Okay, audience, I move to the next slide. Okay, now I come to the exact topic. What is that? Molecular modeling and molecular docking. So as I mentioned the previous slides, for drug designing, we need structural informations of the small molecules. Okay, the structural informations can be taken from the chemical databases. I told you. Suppose the structure informations not available in the chemical databases, then what can we do? Here, the molecular modeling come into the picture. Suppose the structure is not available, right? The structures of the small molecules can be drawn, and the structures of the small molecules can be drawn, and the structures of small molecule can be generated. 2D structures, 3D structures can be generated using the tools such as Argus Lab, Marvin Suit, ChemSketch. Those tools can be used to generate the three-dimensional structures of the small molecules. The generated 3D structures can be viewed and can be analyzed using number of visualization tools like Discovery Studio, Scavenger, PyMol, RASMol, SwissPDB Viewer. The RASMol, SwissPDB Viewer are freely available. The other three tools are commercial. The first three tools are freely available tools. Okay. Then the properties of the small molecules can be predicted using the Swiss ADME. It's a freely online tools. The structures of the small molecules can be generated in different file formats because each tool is compatible to access certain file formats. So you can generate the files in the different formats like mol format, PDB, mol2, SDF, the different file formats we can generate. So what I'm telling this slide, so we have a list of computational tools to generate the three-dimensional structures of the small molecules, as well to analyze the structural properties of the small molecules. Okay. Any clarifications? Okay, let me move on. Okay, now what is the second requirement for drug designing? The structural informations of the macromolecules. Now, as I told you, the structures of the macromolecules are available in the protein data bank. The protein data bank consists of the structures of the macromolecules determined by NMR and X-ray techniques. To date, NMR and X-ray are the only two techniques to elucidate the three-dimensional structures of the macromolecules. So in this slide, I think I'm clear. Okay. So in this slide, I'm showing the three-dimensional structures of BBTI, bovine pancreatic trypsin inhibitor. That is a protein. The BBTI is the first protein for which the structure would be elucidated by using NMR techniques. The structure was determined by Kurt Utrecht and his team, and he received the Nobel Prize for his monumental contributions towards NMR of proteins in the year of 2002 in chemistry. So NMR is one of the two techniques to determine the structure of the macromolecules. So the another technique, this slide, I hope everyone see. In this slide, I'm showing the X-ray structures, the three-dimensional structures of myoglobin and hemoglobin. The myoglobin structure was determined by John Kendrew and his team. And the hemoglobin structure was determined by Max Perutz and his team. And Max Perutz and John Kendrew received the Nobel Prize for their contributions in chemistry in the year of 1962. So what I like to say is these are the only two techniques to date to determine the structures of the macromolecules at atomic level resolutions, at high resolutions. Okay, so now molecular modeling. Come to the picture of molecular modeling. Now for the target you have, the structures not available. The structures not determined by NMR and X-ray. So structures not available in the PDB. So what can we do? Only choice, computational methods. So using the computational methods, the three-dimensional structures of the macromolecules, especially proteins, can be determined. There are a number of computational strategies. As I mentioned in the slide, homology modeling, threading, ab initio. These are the popular computational molecular modeling methods. So using the methods, the three-dimensional structures of the molecules can be predicted. Okay. Now I will explain just, I will give the outline of each method. Okay, homology modeling. The principle of the homology modeling is similar sequences will have similar structures. Okay, so the target is what? Target is the protein for which structure not available. So for the given target, we have to identify the template. Template is the one for which sequence available, structure available. The template and target will have high similarity in the sequences. So using the template structure, the target structure can be determined. Oh, no confusion? Okay. So how to determine? For that, number of molecular modeling softwares are available. As I mentioned on the screen, just you can see Swiss model is an online tool. Modeler is an offline tool. And Scadager is a commercial tool. So I have given just one example in each case. So there are methods to predict the structures of the macromolecule. Each method has its own merits and the limitations. Okay, but we can predict the three-dimensional structures of the macromolecules using the homology modeling. Okay. Now another method is called threading. The principle of threading is dissimilar sequences may have similar structures. Surprising? Dissimilar sequences may have similar structures. So in this case also, we have to identify the template to the target. But in order to identify the template, instead of comparing the amino acid sequences, we will be comparing the properties of the amino acids in target as well as the template. I hope you understood. Okay, so using the properties, we will identify the appropriate template to the target protein. Then using the template, you can predict the three-dimensional structures. This method is called threading or fold recognition. So the another method is called ab initio. So ab initio is a template-free method. The homology modeling and threading are template-dependent method. Ab initio is a template-free method. The method works on the basis of the first principles. However, the method is applicable only for a peptide, not for the proteins. The method is a very early stage, and there are a lot of scopes to improve the ab initio method. And the very one of the important limitation is the structures determined, predicted by the ab initio method cannot be validated in a straightforward manner. Okay, so if you have any questions, I will give more answer later on. Now I just move on. So now what I'm telling here, so the structures of the target molecules can be modeled using the homology modeling, threading, and ab initio. I will give the virtual demo, right, for the homology model. Okay. I think students will be very much interested, I hope. Okay, homology model. So assuming now BCLB, you can see the slide, BCLB. BCLB is the anti-apoptotic protein. H stands for the human. The organism is human. So the BCLB from human has a sequence but doesn't have the structure. So that is our target. So our target doesn't have the structure. So we have to predict the structure of the protein. So for that, what we have to do, we should identify the template first. Okay, the template is identified. Template is BCLB from the mouse. For the BCLB from the mouse, sequence available, structure available. And the BCLB from the mouse and the human are in high similar or high similar in their sequences. So it is a best template. Okay. Then I should have the alignment file, atom file, script file, then structure can be predicted. Okay. Now what is the first question? Sir, how to identify the templates? How to generate these files? I will explain next 2 or 3 minutes. Okay. So students, you have the target protein for which structure not available. So you want to predict the structure. You want to predict the structure using the homology model. How to do it? Okay. Very simple. Just I will explain step by step. You just note down. Go to the NCBI. Go to the NCBI website and select the blast. I have the blast I have circled in the red color on the screen. So select the blast. Then a new window will pop up. So in the new window, you will have the number of blast tools. Select the protein blast. Okay. Now another window will pop up. So in the new window, you just paste your target sequence. The target is the one for which structure need to be determined. So paste the primary structures of the target protein in the text box. Then you select the database protein data bank, because you want to identify the template for which structure is available. So structures available in the PDB. So you want to search the PDB databases. So paste your target sequence and select the PDB database. You can go with the default parameters, then just blast it. You will get the number of hits as I show in the slide. So these are the hits. These are the sequences which are highly similar to your target sequences for which structures are available. So now you have to select the appropriate template. So you can select on the basis of the total score, on the basis of the query coverage, e-value, and the identity. The e-value should be very less, and the query coverage should be more than 90%. And the identity should be higher. The identity is the best one. Higher the score is the best one. So on the basis of the score and other parameters, you can select the template. So now what is the template? 2KUA, that is PDB ID. So template is selected. I hope now you understood how to identify the template. Okay. Now go to the PDB database and download the template structure, atomic file, 2KUA. That is a template file. I have circled in the red color. Okay. Now what you have done, you have your sequence, target sequence in your hand. Now you identify the template. For the template, you have the sequences, you have the structure. Okay. So now do the alignment. You can do the alignment using the multi-align tools. Target sequence, template sequence. You have to paste in the FASTA format, and there are parameters, but you can go with at the beginning. You can go with the default parameters. Just do the alignment, and you will get the sequence alignment sequences. Okay. Now you can save it in the alignment file. Okay. Now you got the template, you got the alignment. Atom file. Atom file is nothing but your PDB file of the template. So atom file also you have. Now what do you need? Script file. Script file, suppose if you use the modular, the modular tool will provide the script file. In the script file, you have to mention what is the name of your target file. What is the name of your template sequence file? The name of the atom file, name of the alignment file, and how many models you want to generate. You can generate 1 model or 10 models as you wish. And the model can be validated by the unique parameters, which is called DOPE, D-O-P-E. Okay. So by fulfilling these parameters, you can run the modeler, and you can get the structure. That's it. So now on the screen, you can see the 2 structures, blue and red. I think it's clear, right? Red color and blue color. The red one is a template structure. The blue one is the modeled structure, the target structure. So using the homology modeling, right, you can predict the three-dimensional structure of the protein molecules. Okay. For that, I told you there are a number of tools available: Swiss model, modular, Schrodinger. Each tool has its own limitations and merits. So you have to understand what are the merits and limitations. Accordingly, you have to use the tool. But generally, modular will be okay for most of the cases. Schrodinger is a commercial one. Okay. Okay. So this is what the molecular modeling of the macromolecules. Any questions? If you have any questions, you can unmute and you can ask me, no problem.
You have any doubt, delegates? No, sir, you can proceed, sir.
Okay. Now we know to some extent. Okay. Now you know how to model the small molecules, how to model the macromolecules. So what is the next step? You want to see how the small molecules interacting with the macromolecules. That is coming the topic of molecular docking. So in the molecular docking, you can make the complexes of macromolecules between the two macromolecules or macromolecule and small molecules. So the number of molecular docking tools are available. So I have mentioned a few of them here. Autodock. So Autodock is a very popular molecular docking simulation tool. It's free. It's freely available. It works Windows as well as Unix. And CSAR. CSAR is a commercial tool. It's a fantastic tool. I'm very happy to say we have in our laboratory. It's a molecular modeling, molecular docking tools. The StarDrop. StarDrop is a PKPD estimation. The tool is very useful for estimation of PKPD values. GROMACS for the molecular dynamics. I will be talking about each tool, in a brief. Let us start with the Autodock. So these are the molecular docking simulation tools. Okay. Actually, I am going to present the interactions between anti-apoptotic protein and anti-cancer compound. So just for sake of clarity, just one slide I have about cancer. As I mentioned, my computational designing for the cancer biology. So just I have taken one example from the work I have done. See, as we know that cancers is uncontrolled cell proliferation, right, due to genetic disorder and the impairment of apoptosis. So my lab is mainly concentrating on the apoptosis. So apoptosis, as you know, is a programmed cell death, and in the cancer cells, the anti-apoptotic proteins are overexpressed. So we need inhibitor molecules. So if you could design a very good inhibitor to the anti-apoptotic protein, in other words, we are designing the anti-cancer compounds. Okay. Let me move on. Okay. So don't worry about this molecular structure. Look at the structure of XJ, the two molecules on the bottom of the slide, XJO34C, XJO35B. Okay, just to say XJ34, XJ35 for sake of clarity. So look at the molecules. Anything interesting? I have just a break.
Staff members also there.
You find any interesting observations between the XJ34 and the XJ35? So look at the molecule, just look at it. So very, very interesting molecules. That's why I'm asking the question. See, in the XJ34, right, the two molecules are very similar. Two molecules are very, very similar. The XJ34, we have the two methoxy groups at the position number 2 and 3. In the case of XJ35, the two methoxy groups are present in the position of 3 and 4. So in the case of 34, 2 and 3, the case of 35, 3 and 4, except that there is no difference at all. So the 34 and 35, XJ34 and 35 are structural isomers. Okay. So what is so interesting? The XJ34 is anti-cancer compound. XJ35 is not an anti-cancer compound. Okay. So how we can relate to our studies? See, experimentally they have shown one ligand is interacting with the anti-apoptotic protein, which one XJ34 is interacting with the anti-apoptotic protein and inhibits the activity, so it is anti-cancer compound. But 35 is not interacting. They have shown by experimental methods. But what is the mechanism? Mechanism by which the 34 is interacting, 35 is interacting, cannot be brought out by experimental methods. Very, very important. Because when the molecule is not interacting, it cannot be characterized by experimental methods. Here, the molecular docking come to the picture. So I have taken one very, very unique example for you for the presentation. So one molecule is interacting, one molecule is not interacting. The two molecules are highly similar. So when they are structurally similar, how it can be differing, how it can be differed in their structure, in their biological properties. Okay. To answer this question, we are going to use the molecular docking simulations. Next 2 slides, I'm going to show some very hi-fi experimental techniques just to make you understanding, yes, one ligand is interacting, one ligand is not interacting. In other words, to convince you, because experimental data is more reliable than the computational data, of course. But computational data has its unique strength to address some of the questions that cannot be answered by experimental methods, as well to speed up the experimental methods. Okay. So to convince the audience, I'm showing the another 2 slides. The data, okay, may not be so familiar for you. Okay, before that, okay, here I'm showing this slide. What I'm showing is the structure of the anti-apoptotic protein. As you can see, the anti-apoptotic protein is a BCL cell, and it has 2 surface grooves. I have labeled BH groove and the BH3 binding groove. The small molecules that are capable of interacting on the BH3 binding groove can act as an anti-cancer compound. Very simple. So the molecules that are interacting in the BH3 groove will inhibit the activity of the anti-apoptotic proteins. So the compound can act as an anti-cancer compound. Okay. So now the focus on the BH3 binding groove. Okay. Just 2 slides. I'm showing some very advanced experimental methods to show that one ligand is interacting, another ligand is not interacting. Don't bother. If you are not understanding, it's okay. You can ask. If you have any question, you can ask me also. Otherwise, just understand. Yes, this ligand is interacting for that purpose only.
Once again, sir. Kavita madam has asked some question. Once again, sir.
Okay, okay, sure.
How it is both are ortho position only no. How to know interaction, sir? Better Kavita madam can unmute yourself and then ask now.
Yes, yes, that is better.
Kavita madam.
Please ask the questions, please.
Yeah. Can
Sir?
unmute yourself. Yeah.
Yes, sir. Yes.
Sir, in this, yes, sir. You said no, sir, that 34C and 35B methoxy group are 2 molecules are having that ortho position only, no, sir. So it is not only. Yes. So how you said no, the ligand interaction it will get differ ligand interaction. So that is my question, sir. All molecular formula same only. Both are in ortho position only. So just I have a doubt. That's why, sir, I asked.
Okay. So let me understand your question very clearly. So 2 molecules are very similar.
Yes, sir.
Same molecular formula, but why they are differing in their biological properties? Is it your question?
Yeah, yes, sir.
Okay, that I'm going to answer. Okay, okay. So, see, look at the structure of the 34 and 35. Same molecular formula. So it's a structural isomer. the position only differing. The methoxy group in the 34. So you have to consider the 6-membered ring. The position where the 6-membered ring is joined is the position number 1. So
Okay.
the two methoxy groups are attached in the position number 2 and 3 in the case of XJ34. In the case of XJ35, it is located under 3 and 4, except that there is no difference at all.
Yes, sir.
Yes, sir. Okay, so the two ligands can be called as structural isomers.
Okay.
Generally, what we think, when the two structures are very similar, we expect the biological activity also very similar.
No, okay,
Yeah,
sir.
but,
No, sir,
but
that is not my question. Actually, biological activity, it will be, I'm asking, you said no ligand interaction. Somewhat less it will be, but biological activity, it will be no, sir. Both
okay,
structure.
I will say, I will
Okay.
explain this.
Okay. Okay.
Mm.
Okay, sir.
Okay.
Thank you.
Okay, my
Yes,
pleasure.
can you unmute it? Oh.
Okay, I will address the questions very clearly now. I have all the slides, I hope. Okay, here I'm showing, this is the NMR, two-dimensional NMR spectra. This spectra is called HSQC, nitrogen 15 HSQC. You can see number of spots. See, I don't know how many of you are very familiar with that. The technique is very simple, but you should have the basic knowledge about the NMR, that's it. So now, each spot represents the amide protons. So you have the red spots and blue spots. So red spots represent the amide protons of the BCLXL protein alone. The blue is the blue spots represent the amide protons of the BCLXL treated with the ligand XJ34 at 1:3 ratio. So I have overlapped the two spectrums. Okay, the BCLXL alone and the BCLXL treated with the ligand. Now, based on the chemical shift perturbation, we can make sure the ligand is specifically interacting with the protein or not. Right. So, based on the chemical shift perturbation, so a set of residues will show a chemical shift perturbation in the spectrum. For example, you see the spectrum. You can see on the top. You see on the top like C148. I just tell the number of the residue: 148. 147, 196, and 109, and you can see some residues in the bottom, M218. Okay, so these residues show some chemical shift perturbations. Very important thing is all residues are present. Only a set of residues shows chemical shift perturbation. So when you have all the residues as well as a set of perturbation, we can get the idea that the ligand is probably interacting with the protein. Okay. Now let's see what happens with the 35. Here I'm showing again the same kind of experiment, protein with the ligand at a different ratio, 1:1. The first one is 1:1, second 1:3, 1:5, 1:7, and 1:10. But unlike in the previous slide, this slide, you see one remarkable difference, right? So you look at the blue spots. The blue spots get clumped. And means what? The protein undergoes some drastic structural changes. The drastic structural changes is a denaturation process, not the binding process. If it is binding process, you will get the distinct peaks in the NMR. When you have the clumped peaks, it means that the protein is denatured. So it shows that the ligand is not interacting with the protein. Okay. So this is at the residue level. I'm talking about at the residue levels. One ligand is interacting, one ligand is not interacting. Okay. Now I'm showing at the molecular level, because I know this problem is very unique. It will bring a lot of skeptical. So that's why I told you very beginning itself, I want to convince the audience first. So I'm showing experimental methods. Yes, see, one is interacting, one is not interacting. Right? Even the madam asked me, right? Definitely there will be an interaction, sir. Okay. I want to make sure. See the interaction here. Here I'm showing the ITC experiment, isothermal titration calorimetry experiment. It's a very beautiful, excellent techniques to make sure the the binding efficiency of the ligand with the protein. To my knowledge, that is the best technique. So in the top panel, you can see the XJ34. Each spike represents the amount of the heat released upon adding the ligand to the protein. Okay, and the bottom panel, the area under the peak, area under the spike is integrated. Now we have the data. The data can be fitted to an appropriate equation, which can yield the binding constant. So what is the binding constant for the BCLXL and XJ34? I have mentioned on the slide. You can see 3.4 micromolar. It's a very appreciable binding. 3.4 micromolar. Nanomolar is really great, but 3.4 micromolar is very near to the nanomolar. That shows the ligand is very high affinity to the protein. Okay. Now look at the 35. BCLXL and 35. The same experiment. I need not explain again. And what is the binding constant? 750 micromolar. So 750 when you have the binding constant 500 or above 500, generally we say it's a non-specific. Any molecule you take and you add to the protein, it shows some kind of binding. That means it's not binding, it's a non-specific. So binding means the structure should not be disturbed. The ligand should exactly fit into the pocket and it should inhibit the activity without destroying the structure of the macromolecules. Now look at the 35, the binding constant is 750. 750 means it's non-specific. So I have so many experiments, but this is computational presentation. So just to convince you, I showed two best experimental reports at atomic level resolutions and molecular level resolution. So what you understand, 34 is interacting. 35 is not specifically interacting. That's very, very important. Specificity is very important. Okay, you can take any small molecule, macromolecule you add, there may be some interactions, some electrostatic interaction. Doesn't mean that they are interacting. The interactions should bring some positive effect. Activity should be increased or activity should be decreased. That's very, very important. Okay, now, so these are the experimental data. Now, how to bring the answer? Why, when the structures are similar, one is interacting, one is not interacting? So experimentally cannot be proven because one is not at all interacting. So now computational come into the picture, molecular docking. So now I have generated the complex between the 34 BCLXL and 34. I'm showing the complex over here. So I can finish it in another 10 or 15 minutes. Don't worry. So this is the complex of BCLXL and XJ34. Okay, now I generated the complex using the autodock, BCLXL and the 35. Surprising? So I have shown, see, the 34 is interacting in the BH3 binding group, right? BH3 binding group is very, very important, where the ligand is docking. Okay, now look at the 35, the same position, the ligand is docking. So what does it mean? That means the computational method is wrong, because experimentally the ligand is not interacting. But computationally I am showing both ligands are in the same pocket. So how to answer? Okay, so you have to see the binding energy. The binding energy for 34 is appreciable. The binding energy for 35 is not appreciable. But it shows the same binding site. Then, because the computational studies with some violations, the interactions between the two molecules can be shown. But we have to be very smart to analyze those interactions. The interactions are very unique or violations, and those parameters need to be addressed using the various strategies. Okay, that I will explain. Okay, now look at the two molecules. I have overlapped the 34 and 35. I'm showing on the BH3 binding group. Look at this docking simulations, very perfect. See, the two molecules are perfectly overlapped. It's two different experiments. And now I have overlapped the structures. The magenta color is the XJ34 and the cyan color is XJ35. Both are in the same binding group. Poses are very similar. Okay. Now look at the slide. Now I have colored. I have standard color of the residues which are in the 4 angstrom proximity to the ligands. Okay, I'm showing the front view as well as the rear view. So you can see the position of the ligands. The 34 is perfectly fit into the group. Perfectly fit into the group, within the group. But 35, look at the 35. One group, what is the group? Methoxy group is exposed to the negative environment. And you can see the front view as well as the rear view. I hope you can see the rear view also. Right? You can, I think students are very smart. You can orient your what is called the cell phone in the horizontal positions, so you can see the picture in the zoomed view. Okay, so you can see the methoxy group exposed to the negative charged environment. Okay, so what is the point over here? The methoxy is a very simple chemistry. So the methoxy is a negative charged. Now the methoxy group is exposed to the negative charged environment. So there is a charge incompatibility. With the violations, the computational method, we can show the binding interaction with a very, very poor binding energy. Okay. But now what will happen? Because of the charge compatibility, in reality, what happens? The methoxy group will try to move within the BH3 binding group. Okay. Now the methoxy group is attached to the 6-membered ring. That 6-membered ring is attached to the another 6-membered ring. Now another very simple chemistry. When the two 6-membered rings are joined by a single bond, one of them will be perpendicular to another one. You can see the structure very clearly. One ring is vertical, another ring is horizontal. Okay. Now because of charge incompatibility, the methoxy groups move to the BH3 group. When it moves, the ring structure changes. Accordingly, the connected ring structure will change. Accordingly, the rest of the structure will change. So the resultant structure, right? The resultant structure is incompatible to fit into the groove. That's it. See, very simple. Because of charge incompatibility, the methoxy group is the culprit. The methoxy groups try to come within the groove. Because of that, the whole structure of the ligand get into the different conformation. That conformation cannot be fitted into the groove. Because not only the chemical complementarity, the structural complementarity also very, very important to fit into the group. Because of that position, methoxy groups, the ligand is not interacting. These informations we are getting only from the computational methods. And we have published. We have published in the Journal of Medicinal Chemistry. In fact, it's about 7. So it's convinced the international reviewers. So this is not so new, but case to case is different. For example, you see the estrogen and testosterone. They are differing by just only one group, but one is the female hormone, another one is the male hormone. Okay, you need not to go even that much. Take the ribose sugar and deoxyribose sugar. Only one group is different, but ribose sugar is in RNA, deoxyribose in DNA. Ribose sugar never come to the DNA, deoxyribose never go to the RNA. Just one group difference. So what I'm telling is, it's a very important point. Right? There is a common myth. Structurally similar ligand will have the structural similar properties. I agree, but it cannot be taken granted always. Right? And case to case is different. When we have that kind of thing, don't think it's a problem. It's an opportunity for us to understand the mechanisms by which we can design the ligand. So using that property, we have designed so many ligands, and there are something in different stages, paper publication and patents. Okay, I hope now it's very clear. Sir, Sandil sir. Sandil sir. Sir, can we move on, or is the time's up? So what can I do? I can stop, or I can continue another.
Yeah. You can continue, sir. You can continue.
Okay, sure. Okay, sure. Okay, now another tool, CSR tool. I will finish in another 10 minutes. Don't worry, audience. I hope this talk is very interesting. I believe. Okay, next 10 minutes we will wind up the session. CSAR, the CSAR is a commercial tool, is available in our laboratory. Okay, it's a fantastic tool. I have some kind of work with your professor, Dr. Sendil Kumar also. Okay, see here, so I have the ligands. For the ligands, I can get the structure from the databases. Okay, so I go little fast and look at the picture. The tool is, provides the binding energy, the binding score, and the binding affinity, as well as the poses. So I have did the docking, and I'm showing just one protein ligand poses. Okay, then this is the 2D picture. What you can see, once you have generated the complex, you can see residues which are interacting with your ligand, and what type of interactions. There are many type of non-bonding interactions: hydrogen bond, electrostatic, hydrophobic, van der Waals interactions. Those can be analyzed. And here, I think everyone see the slide. Based on the binding energy and the ligand efficiency, we can sort out the best ligand, showing the affinity to the target molecules. Okay, so you can see the binding energy. I have selected the 2 compounds because those are high in energy in terms of negative sign. Okay, in this picture, what I'm showing, so I have taken one ligand on the protein structures, right? I am maintaining the core, because core region I'm maintaining, and I want to design the ligands. Now from the molecular modeling, molecular docking, I move on to the drug design. So by maintaining the core regions, I want to add different portions in order to get the best binding efficiency to the target molecules. That can be done by CSAR method. So I don't know whether you are capable of seeing the cursors. There is a cursor in purple colors. So that portion I'm removing on the top and bottom, and I'm generating so many new molecules, right? Then I do the screening. I do the screening for the newly generated molecules, right? And these are the generated molecules. So these are the designed compounds. Designed compound 1 and designed compound 2. This shows much better interaction than the parent compounds. So what is the message over here? So you can do the molecular docking, and based on the molecular docking interactions, we can design the compound also to improve the affinity. I hope. Okay, let's move on. Okay, now I come to the star drop. Star drop is a tool which can be used to predict the PK/PD properties. There are tons of properties can be predicted. So I have mentioned a few of them. You can take the screenshot if you want, and you can think about. Okay, so I think most of the properties, as a pharmacy students, you know very well. BBB as a blood-brain barrier, HIA human intestinal absorptions, and efflux parameters, and some kind of inhibitors, and other structural parameters you can see here. Okay, all the properties can be predicted. Okay, so on the not only on the basis of the affinity, on the basis of the PK/PD values, we can we can identify the best ligand molecules. Okay, now you identify the ligand molecules. Assuming that the ligand molecules not commercially available, what can we do? So we have to synthesize. Our tool, the star drop tool, also will be very useful to sketch the path, the chemical synthesis path. So there is options for that, retrosynthesis, and for the given compound, we'll give the best chemical synthesizing protocols. The tool provides the chemical synthesizing protocol also. So the compound can be synthesized. Okay, so this is another very important properties. Suppose, for example, as a pharmacy students, you know very well about the Lipinski rule. Okay, the molecular weight should not be greater than 500. Number of hydrogen bond donors should be less than 5. Acceptor should be less than 10, and log p value should be less than 5. But you know the rules for what? Rules for making the good drug, right? But so what you can do, you can give the weightage. You can give the weightage for the each parameter, and using the weightage on the basis of the data you have. The weightage you are giving and the weightage I am giving may differ, and I will have my own rationalization, and you may have your own rationalization. Accordingly, the molecule can be identified. So giving the weightage, we can give this, we can calculate the score. This is called multi-parameter optimization. It's another fantastic models we have in the CSAR, sorry, star drop. Using that, we can identify the drug molecules. Okay, if you have any questions, I will explain more. Now I come to the another part, the molecular dynamics. Okay, so the molecular dynamics is what you can do the molecular dynamics for the protein alone, or protein ligand, or protein-protein, protein lipids, and time-dependent, temperature-dependent molecular dynamics, and the steered molecular dynamics. Anything can be done. The molecular dynamics also very important to study the interactions as well as to understand the stability of the protein. I will show one simulation. It's the last slide. Here I'm showing, I hope everyone see the movies, it's a molecular dynamics. So here I am simulated the interactions between the protein and the erythrocyte membrane. So what is again here interesting, studying the interaction between the protein and membrane is an uphill task. It's very, very difficult by experimental methods. So where the molecular, the computational biology come to the picture. So in the computational methods, we can study the interactions between the protein and the membrane. This membrane is a heterogeneous. So heterogeneous molecule cannot be studied by experimental methods, biophysical experimental methods. So now this is a protein, this is the snake venom protein. And the snake venom protein is known to lyse the erythrocytes. So now we want to see how the protein and the erythrocytes are interacting. So you can see the beautiful movie, right? The protein, it has 3 loops, and one of the loop is interacting. The middle loop is interacting with the membrane. And there are a lot of, it's not biased at all. So we we change the orientation of the protein in different level, at different distance, and we do the comprehensive analysis. Then we come up with the part which is interacting with the erythrocyte membrane. Accordingly, we design the ligand. That ligand is inhibiting the protein. So that ligand is antivenom compounds. Okay. So the logic, I hope you understood. So I stop the movie over here. Okay. Now I come to the concluding remarks. So what I mentioned, I just read the concluding. Okay, I put the zoom. So the computational tools, right, can be successfully used to screen the lead compounds and also designing the de novo small chemical molecules. The in silico tools are indispensable to efficiently speed up the drug designing process. Okay. So computation tools is very important to speed up the drug designing process. It's very important to screen the lead compounds as well to design the compounds. There are 4 screening strategies. I told you all the screening strategies can be carried out by computational methods. And very important point I would like to make from the studies to the smart students, just to make sure in mind, structurally similar ligand may not exhibit similar binding affinity to the receptor protein. The concept is good, valid, but case to case is different. So keep in mind that structurally similar ligand need not exhibit the similar binding affinity. That is a take-home message I want to make it from this presentation. That is a unique point. I hope you can keep it in mind. Similarly, structurally similar proteins may not exhibit similar binding affinities. That is a different story.